Alrighty, let's see if that works. Ooh, I think we're good. I think we're live. Okay, well, hello everybody. Welcome to Fantastic Microbes and Where to Find Them, your window to the microscopic world. And today is our second live stream ever. So very exciting, very cool. We had enough people last time that it seemed like this would be a good idea to keep doing. So if you guys are interested, this is definitely all, brand new for me. So we'll kind of see how it goes. Hi, Oliver. Hi, Bite-Sized Stories, if you're still here. I think you commented a little bit earlier. Yeah, glad to have you guys here. Let me know if the music is too loud or too quiet, or if you even hear it at all. Again, don't really care too much about the music, but it kind of helps with the vibe, I guess. Yeah, hope you all were having a good weekend and week, I guess. Now that we're in, I guess it's Tuesday, we're almost into the middle of the week, so yeah, very exciting stuff. I have a note that I was going to say earlier, or like before we get started, and it is that I have a Discord that I am starting. I'll put the link in this video after I'm finished with it, and then I'll start putting the links in the videos from here on out. And the reason I'm starting the Discord is basically I want to make sure that you guys are all up to date and up to speed when I do live streams, and like if I change schedules, and you guys can give me more input and stuff like that on things that you want. So again, I'll have that link out there eventually. So until then, let's go ahead and just do some microbe stuff. So I have a couple of things that I wanted to look at today. So one, if you were here last week, we looked at a bunch of different samples. We looked at some Great Salt Lake samples, and then we looked at some stuff that I collected near Utah Lake in the Jordan River area. Now, in that specific area, we only looked at 2 drops of water, 2 little microscope slides, and I have these big containers that I feel like there's just more stuff in there. So I wanted to look at that stuff and see, what more interesting things we could find. And then I also am in the middle of filming a new episode for my Small Adventure Saturday series. It's coming out hopefully this Saturday. Where I think 2 weeks ago, we filmed in the, we looked at grass under the microscope, but this time, I wanted to dig a little bit deeper, go right underneath the grass, and get all of the dirt and stuff that is under the grass, because, that's where definitely we'll probably find a lot of nematodes and some other worms, and maybe some amoebas, tardigrades, all sorts of other stuff. So I have a couple of those things, but first, and hopefully stentors. Stentors would be very cool as well. So what are we looking at here? Well, if you saw the description, you probably saw that I was wanting to look at some butterfly wings under the microscope, and I got a collection of wings here just from some random person on Etsy. They said that they were sustainably harvested and collected, so we're assuming that they lived a full and complete life, hopefully. And yeah, so let's just look at this. So the very first wing that we're looking at, it looks like it's very brown, but that is actually not the true color. So the true color of this is going to change. So what I'm going to do here is I have an extra screen. As you'll see here, I have my microscope with the light on. We're going to turn this light off and then switch to, I have this book light right here, and this reveals the true color of this butterfly wing. So this is and here, let me get this big thing out of the way, and I'll just close that. So this is a blue morpho butterfly wing, and they're very common in butterfly collections, and yeah, they're pretty cool. So the unique thing about butterfly wings is that they're a little bit different than like dragonfly wings and like fly wings, insect wings, which usually they just look like stained glass almost, you know, just like really clear, and then like a couple of little veins here and there. Now, butterfly wings do have those, but they're also covered in these scales. So right now I'm at 40 times magnification. That's the lowest one that I've got. Let's zoom in just a little bit more to get some detail here. And this is kind of on a slope, so let me get to a good spot where things are a little bit more flat. And let's see. Yeah, so here we've got some butterfly wing scales all close up. And yeah, very, very interesting. So butterfly, yes, very, very cool. The scientific name for butterfly is Lepidoptera, which I think literally translates to scaly wing. Some really smart guy told me that once. His name's Ian. If you're out there, Ian, I remembered what they were called. So yeah, here they are. I just love how they're just like so nicely lined up. You can see one kind of in the middle here that is like poking up. One kind of interesting thing is that they're only reflective on like a certain side. Did I collect this sample? I collected it by ordering it on Etsy. So I just like Googled or I searched butterfly wings on Etsy. These are tropical butterfly wings. I think these ones are like native to South America or something. So no, unfortunately I did not collect these on my own. But so right here I am just moving the what's it called? Let me show you guys here. So I'm just kind of like moving the booklight, and I guess it doesn't do much good because you don't see the booklight on that side. But let me just kind of show you how it's set up. I basically have it like this. And, you know, if I light it from this side, it's not as reflective. It's almost transparent on this side. So you gotta just kind of light it just right. So I'll put it back over here. And this is kind of an important lesson that I wanted to share, because this whole idea about light changing how things look goes beyond butterfly wings, and it is really, really important in the microscopic world. A lot of the time you'll see like a stentor with like one filter, and then you'll see it with another filter, and one filter, like Darkfield filters, for some reason can turn certain stentors pink. But if you take the filter off, then they'll be like kind of like a bluish green color, and it confused the heck out of me at first. But then I realized that just when light hits these small organisms, it treats them very differently depending on where it hits them. So there's me just adjusting that one. Now I want to take a look at a couple more, and then we'll go into some live samples. So just as people are trickling in, again, so this, let me go big real quick here, and I'll hide this guy. So this is the blue morpho wing, and as you can see, a lot of the scales have kind of fallen off here, and it actually looks totally brown and different on the other side. So very, very interesting. Some of these butterflies, well, some of these wings are actually from moths. Now, I don't remember too much about what the names were of each one. So, anyone here is Massachusetts area, stentors are everywhere. Ooh, sweet. I'd love to see some stentors. Yeah, hopefully we can see some stentors today. Yeah, I'd be kind of interested to know where is everybody from that's tuning in today. I'm based out of Utah right now, and I grew up in Arizona. So, in case anyone was wondering, let's get this dude out of the way here. And okay, so let's see if I can find like the perfect angle where things are really reflective here. Okay, here we go. So, and then I'll adjust the focus and make it clear. Oh yeah, so I really like this one. I made a video about this butterfly wings under the microscope like a year or two ago, like right when I was starting off this channel. This was one of the very first things I looked at. So if you recall, when I was showing you guys the actual wing, some of it was green and some of it was black. So the black, it looks like, doesn't have hardly any reflection at all, and then the green, or like the greenish yellow, it is very reflective and it looks a lot more curved than the other ones that we were looking at. So let me just toy around with this light a little bit more, and let's see, let's zoom in, have some fun zooming in. Yeah, okay, so here we're on the black, and here we have, man, isn't that beautiful? If this was a real recording, unfortunately, like with live streams, I think it kind of like tones down the quality a little bit. But like this stuff in 4K is like really, really cool. I think we're probably streaming at like 1080p or something like that. I don't know. I forgot to check. I usually try to stream at the best thing. Yeah, so here I am just kind of adjusting the light a little bit more. Very cool stuff. Yeah, right? Isn't it cool? I could look at it all day as well. So yeah, I think maybe it'd be a cool idea to, because there's a lot of wings in here, and I think in my original video I only did like 5 or 6 minutes of footage, but you know, with all of the lighting, I could probably get like a really good angle, or like 50 different angles of each wing, you know, and make just like a really long video, like with some sweet music in the background. I should look at blood sometime in a live stream. Ooh, yeah, that is a good idea. In fact, one of the things that's holding up my microscope right now is a box of lancets, so yeah, I just need the little poking device, and I'll poke myself. I guess I could poke myself in another way too, but yeah, blood in the live stream. I'll do it for next week. Just remind me. I can remember that. I'll remind myself. And so I wonder if I can go in even a little bit closer. So let's check out my highest. Oh, now you can't even see it. So the problem with this highest magnification is that my light can't even get through. So I can try the backlight. Okay. So this is the highest magnification that my microscope goes. It's 400 times. And you can see the scales really well. Let me just dim this light a little bit. Yeah, it's too grainy with this one. So let's kind of go back. I think this is kind of the way to go. Oh yeah, so this is what it looks like with the book light and the bottom light. So I can turn the bottom light up, turn it down. Maybe for the right specimen, it would look really cool. When people are like photographing 50 angles of butterfly wing, yes. Sorry, I need to like remember to look back at the chat so like I'm on the same pace as you guys. So it's late, gotta go have fun. Thanks, Oliver. Thanks for showing up, and thanks for being part of the journey. Yeah, what was I saying? I can't remember. How about this? So, yeah, I think I was talking about getting like the right angles for the right everything. These almost look like Christmas ribbons, you know? And like I feel like this would be like a rooftop at Santa's workshop or something. Very cool. Now, I could look at this all day, as many of you guys probably could as well, but, you know, we've got plenty of other stuff to look at. So let me get this one out of the way. Again, you can see here's the one that we were looking at right there. You can see the black and the green. Next up, this one I just want to see if it has kind of like the same texture. So this one is kind of black and green as well, but it's not as shiny as this one. It's kind of hard to see because this is backlit right here, but this one right here is a lot more shiny than there it goes, than this one. So I'm going to put this one under and see what happens. So let's take a look. Is that the black? Let's see a little area of black and a little area of the light blue. Give me just a minute to get the right focus. And so, okay, so that light's off. Okay, so this one, I feel like the scales aren't as like curved as the other one was. So, and I don't know if it's just the way that the light is hitting this, but it seems like the black scales behave a little bit different than the blue scales. So let's zoom in and get a closer look at this. Oh no! Oh, there we go. Let's see. Okay, so here's our black scales. And again, this is kind of at an angle, so only part of it is in focus right now. But, yeah, what do they taste like? That is a good question. I should call in Mitch from the Water Jet channel and have him do the taste test. If any of you guys have come over to this channel from the Water Jet channel, I did a collaboration with them about a year and a half ago, and that is still one of my big sources of people. So, but I love finding out where people find me. Here, this is the blue section again. It does look very delicious. And, yeah. You know, I bet it probably doesn't have that much of a taste. I actually am kind of curious right now, but this whole thing cost me probably like $10, and I only have so many of each color, so I don't know, maybe if I get like 50 likes on this video, then I'll eat one of the butterfly wings for the next one. How about? Or maybe not 50, but that's too low. Maybe 100. Okay, 100 likes on this video and I'll eat a butterfly wing. There you go. I don't know. I'm like trying to think of like incentives for this channel of like, you do this cool thing and then I'll do this cool thing. I don't know. Oh, this is kind of cool. So with with insect wings and with butterfly wings, I mean butterflies are insects, but like, you know, like how in regular wings, like there are these veins that kind of like go throughout the whole wing. So with butterfly wings, those veins are still there, but they have like an extra, I don't know if it's like an extra layer of protection or if it's just like more three-dimensional because they are like actually 3D. I don't know, I'll have to see. That's a good question for future Jordan. So, okay, this was a good one. Now I want to do one more butterfly wing, and then, like I said, we'll go do the live samples. So, again, here, let's do Show Big Jordan right here. So this is the final wing that we're going to look at. Lots of different colors. As you can see, I kind of damaged it a little bit through just the last video that I made like a year ago. Can they regrow these flakes? That's a good question. I don't know. I know that like butterflies like live a pretty short life, like once they reach their adult stage, so I wouldn't imagine most of their, or like any of their energy would go towards that. That's just my reasoning behind my guess that they probably don't. But maybe they do. Maybe they do. If somebody wants to Google that, go ahead. I think this one is a moth wing, but I can't remember. So I have to go back to this Etsy website and see who I ordered this from, and then see if they have the names of these. Yeah. So this one is, you know, multicolored. You can see it's got some of the black ones. There's some yellow. Lots of yellow and black over there. Let me scroll over. Here we've got some cool green, like bluish, greenish, white. And I hope this is like coming up at least a little bit clear for you guys. Again, still trying things off with live streams, and if the quality works well or not. Oh, now I remember what I was going to say earlier. So with lighting, you know, like how I said earlier, so this is all being lit from the top with just like a book light. Sometimes I've used like my phone light or my wife's phone light while I record with the other one, and those make really great photos. But one of the things that professional microscope photographers do is they'll have multiple lights like that for taking pictures of insects and stuff. Like, you know, like I guess butterflies, or like dragonfly eyeballs. Like, have you ever seen like, oh, sorry, there's some loud cars out there. I don't know if you can hear that. There's like those scientific magazines that have like really big close-ups of bugs with their compound eyes. Those are usually taken with, you know, a compound microscope, and they are like, there's several different photos taken because it's really difficult to get everything in focus all at once. I mean, for example, this butterfly wing is mostly flat, but it can still only be in focus one bit at a time. So like I can focus on just like this little line right here, or I can like adjust the focus and focus on some lower lines. And so what photographers do is they take, you know, 50 different photos, and they just change the focus a bunch of times, and then they just stack all of them together. There's a couple of softwares that you can do that with. And there's another microscope YouTuber, his name's Microbe Hunter. He just did a really great live stream on how to do that kind of photography and photo stacking stuff. He gave a great tutorial on that. Let's move on just a little bit here. I just want to make sure that we cover all of the colors on this. So some of this is like yellow, some of it is a little bit more gold. Google says they cannot regrow the flakes. Well, that is sad. I'm sorry, butterflies. You know, there's actually like some really cool images that you can see on Google if you're looking at butterfly wings under the microscope. Scientists have put this under like a scanning electron microscope, which is like, you know, $100,000 cooler than mine. And with those, you know, you're seeing things extremely, extremely close up. And so you can see really, really cool, crisp detail. So this is kind of towards the end of the wing. These ones are more folded over, I guess, which is kind of interesting because I thought that they were shinier. And so maybe the fact that they curve means that they're able to shine in more places. Yeah. Very interesting. Sorry, I'm just kind of fixing the light, seeing if we can get a decent thing there. There we go. Yeah, man, butterfly wings. Pretty cool stuff. But yeah, so maybe you can see this, maybe you can't, but on some of these wings you can see, like on these scales, there are these lines, and with these lines in the scanning electron microscope, you can see that it's almost like a net pattern. It's very, very cool, so definitely worth looking at. And oh, this is kind of interesting. So at the corner of the wing here, there are a bunch of little hairs. So I don't know what the purpose of those hairs are. Let me zoom out so you can maybe see a little bit better. Yeah, here are just some of the hairs. My light is kind of weird on it, but plus it's kind of tricky to focus all at once. Yeah, this is all kind of curved, and so it's hard to get everything in focus. You guys don't want that for the entire live stream, do ya? Everything out of focus? No, we don't. Show us the live stuff. Yeah, I don't really know what you guys are, you know, mostly interested in. So kind of the purpose of these, like, especially in the beginning with these live streams is, you know, I said last week, you know, I'd show some prepared slides and stuff, and I do have like a bunch of non-living things, you know, like these butterfly wings. We could do like blood next time, and you know, I'll take your guys' suggestions. I think with the microscopic world, most people don't know what looks cool and what doesn't look cool. So, but if anyone has ideas of what they want me to look at, go ahead and let me know. We've already covered bodily fluids. We've talked about it before. We'll get there eventually. But yeah, there's so much stuff out there. So okay, we are at about 30 minutes in, so I think it's time to start looking at some live stuff. So here, let me switch cameras here and change to this. This will kind of be like the big one, and we'll go black on that so you can kind of see. Yeah, so there was this. There's this. Pretty broken. And yeah, so that's 40 assorted tropical wings for art projects. So I bet if you look that up on Etsy, you can find that. But I bought this a couple years ago, so who knows. Yeah, they almost did look like stadium seats at the end. That was definitely kind of cool. So yeah, here's what I've got. Let me remove this. Sorry, it takes a second to transition here. So again, I was just using this book light right here. It's not very bright, 2 AAA batteries, and as a bonus, it doubles as an actual reading light, which is very cool. Not just for microscopes. Okay. So, here's what we're gonna do. I'm going to go big mode right here, and you'll probably see some weird lag with my computer, so let me just turn that screen. And now, with this camera, look, I've got this camera here, and I can show you the setup right here. So here I have some dirt, water under the microscope. This was collected right underneath some grass, and so I'm just going to, I have a cover slip, or a glass slide right here, my cover slips, and then a little dropper here. So I probably should have like another light over here, but So I'm going to take a couple samples here. I'm going to put them on, I'm going to put 2 on the same slide. And so I'm going to put one kind of on this side and one more on this side. This first one is just some debris that I was able to collect from the bottom. And then with the next one, I might use like some scissors or tweezers to get like some actual dirt or like some plant matter. So just add a couple of drops there. Now those drops have quite a bit of stuff, so I'll add just kind of one more drop of water. Usually with these pipettes that I use, the trick, it's about 3 drops of water that does the trick. Now these are some brand new cover slips, so hopefully they are good quality. Sometimes when you open up some new cover slips, they might all be trashed, which isn't fun. What is that book? Oh, so this is, I'm assuming you're probably talking about this one. This is called The Ancestor's Tale. It's written by Richard Dawkins, and who he's, I guess for the family in my chat, he's so he's a really famous biologist, and he's written lots of books on biology. He is also kind of a big opponent of creationism and stuff. So he's like kind of like a big atheist figure. But that's not why I have the book. And anybody, believe what you want to believe, and whatever helps you live a happy, fulfilling life is what I think you should believe. But anyway, this particular book is it's the story of evolution, but it starts from humans and it works its way backwards to all of our closest relatives, and closest living relatives and closest non-living relatives and stuff. And it is a fascinating book. It was required reading for one of my biology classes, and or no, it wasn't required, it was extra credit. And so I read it, and it's hands down like one of my favorite books about biology in general. And again, it's like Richard Dawkins, he's a little anti-creationist or whatever, so there's like a couple quips about that. So if you're not into that, you can just gloss over those 2 lines, because it's really a biology book. But I just give that all fair warning just because he's kind of well known for those other things. But the reason I use that as my microscope book holder, or my microscope phone mount, is because it's just really wide, and so it's able to hold a lot of things. So let me close that up. Yeah, there's another author who I really like, who has just written a really good biology book that I'm really excited for. This guy, his name is Ed Yong, and he wrote this book called I Contain Multitudes. And it's all about the bacteria living in and around us and stuff like that. Yeah, okay. I think we're done with this camera. Unless you guys want to see the microscope, let me know. If you don't want to see my face, let me know, and I can move that out of the way. I think it makes it a little bit more humanizing to see a human face here. So, okay, let me just adjust some things here. Might just take a second. And, let's see. I mostly say all that stuff because my mom is sometimes in this chat or in my microscope videos, and she's like, Jordan, why are you... I can just see her ranting about how I would have this book as my representative book. because she's just a very stalwart religious person. And yeah, but we're here to talk about microbes, so let's talk about microbes. So yeah, looks like we've got one here. This is some sort of ciliate, and most of the things that we see, you can identify them as ciliates pretty easily. Like paramecium is a kind of ciliate. There's lots of other kinds, but the basic idea of ciliates is that they have tiny little hairs that usually cover most of their body. So let's see. Here we've got another one. Looks like it's a different shape, so probably a different species. And then in the very back, you can see there's tiny little dots. It probably just looks like static to you guys, but it's bacteria. I can zoom in here. Oh yeah, there's lots of bacteria going on. So this is my closest zoom. This is 400 times. So this was the highest that we were looking at with the butterfly wings. And you can see there's a couple of interesting looking ciliates here. I wish you guys could see my mouse. I gotta like have a way to circle things. Yeah, back to microbes and stuff. But yeah, so with these, it doesn't look like they're moving too much. Oh, there they are. So I think some microbes can sleep, but some of them don't. And that might just be Jordan making stuff up. So, anyway, these things are very interesting. They've got, like, it looks like they have hairs of different lengths. It's, I will say again, it's really hard to, you know, zoom and focus on this really high magnification. Mainly because light just behaves differently here. So eventually I'm going to get a different phone that does optical zoom instead of digital zoom, because right now this is digital, so each time we zoom in we lose a little bit of quality. What is this sample? This is some dirt underneath some grass that I got from my front yard. So not too exotic of a place. Sometimes I've gone to some exotic places. I've gone to like some national parks and stuff, and but you don't really need to go far away to find really cool microorganisms. And in fact, most of the microbes that you're seeing on this channel, you'll probably see in your front yards or backyards or nearby water areas. And the reason is because, you know, the wind and rain carries this stuff all over the place, and these things have been around for a very, very long time. So with this, we're looking kind of like at a big chunk of dirt here, and it's kind of interesting how the bacteria is all in a big circle around this. You can kind of see it all the way from this back really far view. Very cool. Let me zoom in on this ciliate up here. So this looks more like a paramecium than those other ones did, and he is probably having the time of his life with all of this bacteria. It's probably feeding on all of that. Yeah, the ring around the dirt, it is very, very cool. I don't know if like they were inside the dirt and I just like broke something, like maybe they were like in a little colony that burst when I put the dirt thing on. But yeah, sorry, one of the things that you'll notice too is that the light, like the white balance kind of changes. There we go, there we go. I'm still kind of getting used to this, this app. So normally when I film for my episodes, yeah, some cyanobacteria as well. Normally when I film for my episodes, I film with this app called ProMovie, or I think it's called like ProMovie Plus or something. But for the live streams, I'm doing this thing called OBS Studio, which is what all the gamers use, and lots of other streamers. So, but in order to do that, I have to film with the OBS Studio app. And so, at least I think I do. So I'm still kind of getting used to things. Yeah, so with this filter, so we're looking at everything in Brightfield right now, and in a second I'll switch over to Darkfield so you can see a little bit of a different view. Yeah, these lines of bacteria are just like everywhere. These very unique. I don't think I've ever seen anything like that. Like where they're just in these big lines. Like, is anybody, I don't know if there's other microbe hunters out on there. It's almost like an ant colony. Why are they in this line? Is that weird to you guys? Maybe it's just weird to me. Maybe nobody's ever experienced that before. Let me switch to Darkfield real quick, see if anything looks different. So here's my Darkfield lens. There's a whole black spot in the middle, and that makes the background black. So for anyone that is new to the Darkfield experience, prepare to be amazed. In my mind, this is like the coolest experience. Sorry, I'm just adjusting some stuff on the camera here. Yeah, so all of these little grains that we're seeing, we are, yeah, it's like an asteroid belt. All these grains, like these are grains of dirt and grains of sand. So, pretty cool stuff. I wonder what that is. That looks like... Oh, that's probably too close to see. This looks like part of a insect. Oh yeah, that's the one weird thing about looking at stuff under the microscope. Sometimes you don't realize that you scooped something up until you do. This definitely looks like some sort of insect, and as you can see, it has some circles inside. Those might be eggs, or there might be something else. So it's hard to know when you're looking at something that's decaying. But yeah, so that's that. Let me just keep zooming over. Can't really see the bacteria too well with these, with the Darkfield. Mainly because they're too small for the light. Whoa, whoa, whoa. Do the focus on this. Focus, man, focus. Yeah, so again, just clumps of dirt. It almost looks like all of the ciliates disappeared, and that is mainly because the ciliates that we were looking at were at a the highest magnification that I have, which doesn't really work with my, what's it called? Oh, you can kind of see the line. Yeah, so you can kind of see the line. It's like kind of like halfway, like sliding through. It's kind of like a Milky Way. And here's that line again of bacteria. Whoa. Okay, so it's hard to focus on everything, but this looks, I mean, it's all blurry, but I mean, just use your imagination. I feel like this is so cool. Like, this almost looks like something out of space, you know, like a James Webb Space Telescope view. Again, this is why I always love the Darkfield, because it's like you're looking through a telescope, but it's even cooler because you can actually see alien life. You know, with the telescope, you never know if you're seeing aliens out there or not, but with a microscope, it's pretty clear. There are plenty of unidentified swimming objects all over the place. Yeah, look at this line of bacteria. What the heck? Oh, you know what? The fact that these ones like to create lines probably can tell us what kind of, like what type of bacteria they are. Different types of bacteria make different shapes with their colonies. So some of them like to make, you know, big long filaments. Others like to make like big blobs that are perfectly round. Others like to make just different shapes. I can't remember what that was called. It was kind of in my it was in my microbiology class, or my microbiology lab. Like, we would grow bacteria and then we had to like describe the shapes that the bacteria made. It's one thing to do it in a lab, but it is another to actually see it in real life, because you kind of forget. Yeah, here, let me just get rid of my face so you guys can enjoy this. Oh, no, not that one. Let me just get rid of my face right there. So now you can just enjoy this really cool shot of this line of bacteria and the celestial-sized objects, probably in their mind, you know, in the background. Very cool. Okay, well, I don't have the patience for this. We gotta see some more stuff, you guys. There's so much more to be seen. Like I said, we have 2 different cover slips on this one slide, so we have to look at both of them. So yeah, let's see. Man, that is so cool, that line of bacteria. Who knew that we would see something cool like that? What a joy. Okay, so close your eyes for just a second, because I'm zooming on by real quick. And if you look at this stuff for too long, it can kind of hurt your eyes. So let's see here. With this one, it looks like there's a lot more ciliates and stuff, but there's also a lot more dirt. Let me just kind of find a good, comfortable spot to sit for a second. Normally what I like to do, like when I'm looking at a new slide, is I kind of like to just comb the slide like back and forth, and eventually, like, I'll see one thing that's cool. Like, if it's really, really amazing, I'll just stop right there. But if it's otherwise kind of boring, I'll move on. Oh no, my Darkfield lens fell out. You know what? Actually, I think we can probably see more detail without the Darkfield lens. So let me put this bright field lens back on, and we can see these guys a little bit better. Oh, and let's switch that to auto and switch the focus to lock the focus down. Yeah, there we go. And let's give these guys a close-up. The other thing too is that like there are so many different ciliates and you know, other organisms, both plant-related organisms and other stuff, that I like to just like record everything and then observe afterward, or like record and then like search what the microbes are afterward. Because it's really cool to like learn interesting facts about ciliates and other organisms like amoebas and things like that. But for me, it's just nice to watch them. You know, I mean, most of the videos that I have, I like fill it to the brim with all the facts that I know. But you guys, like on my 10-minute long videos that I do on the weekends, normally I record for like, or like I'm on the microscope for probably like 2 or 3 hours, and then I have like maybe an hour's worth of footage, and then I cut it down. So with this particular sample, it looks like there's a bunch of bacteria, a couple of crazy fast moving ciliates. I would zoom in, but they would probably get out of focus like immediately. They were probably similar to the ciliates that we were seeing before. I don't know if you guys knew this, but I think the fastest organism in the world is a species of marine plankton. Faster than a cheetah, faster than a fast bird. It's plankton, and the reason is not because they can travel fast as in like miles per hour or like kilometers per hour, like really fast, but they move between different zones in the water column, like 100 meters or like something crazy like that. But like for the scale, like for the size of the organism that they're at and the distance that they travel, like by their size, like they travel, maybe it's not that they were the fastest, but they travel more, like they travel longer than any other organism travels in relation to its size every day, like every single time, like when it's the morning, because they're marine phytoplankton, like they move up towards the sun and then they move away when the sun goes away. Anyway, there's your fact of the day. This looks like a dead worm, or maybe it's not dead. I think it was moving just a teeny bit. Or maybe everything else was just moving around it. So, yeah, here's a paramecium-like ciliate. There's thousands of different species of paramecium, so I could probably be safe in saying that that's what this was. But a lot of microscopic organisms look the same, and so you'd need like DNA to correctly identify them. So this sample isn't quite doing it for me, you guys, I gotta be honest. So what I'm gonna do is what I like to do. We will move on to another sample. Got plenty more to look at here. So let's turn this one off, or turn that one on, go back here, say goodbye to that, and then we'll get really big here. And yeah, so normally, if I am able to, I like to let these guys live out the rest of their life as short as it is, and because then I can just get rid of all of them together at the same time. But I'll get like some water and just kind of rinse this off. If you're doing this, make sure that you use spring water or distilled water. If you use tap water usually has like chlorine and fluorine, like fluoride in it, which will kill the microbes. I mean, if that's your goal, then there you go. Yeah, so there's that. Now I've got a relatively clean slide that I can use again if I dry it off right here. There you go. I don't know anything about this camera that I'm using, so I don't know if it's autofocusing or not. Again, I hope you guys are enjoying this show. Okay, so that was all from sample 1 right here, and that was like totally underneath the grass. Now this one, sample 2, was more like right next to the grass, and there was like some moss and stuff, and so I want to take a look at that. So let's see. I'm going to use a different pipette. I always use a different one for each thing, so not to contaminate any samples. So you can see this water is clearly pretty murky, so I'm going to just do like one. Oh, I forgot to lower the camera, so just use your imagination, everybody. Oh, that's still pretty murky, so let me just distill this a little bit. Okay, well, this one might be a total flop, or it might be a total success. You never know. So, okay, here, got a sample, and we're going to place it underneath the microscope. Ooh, and this one has too much water on it, so if there's ever too much water, you can take a napkin or something like that. Dude, that was way too much. You're doing this live? How embarrassing. Well, this is microscopy. Things are going to get embarrassing. It's just how it is. Oh, you guys aren't seeing what I'm seeing. So let's change that. Goodbye. Microscope setup. Hello, Jordan. Hello, very blurry screen. Now this one looks pretty similar to the last one. If this one is maybe a water bear on sample 2, oh man. Yeah, I could show chlorine killing microbes. Eventually, I could make a video on it. Yeah, I've been planning on doing a video of like antibiotics killing microbes and stuff. The only thing is, like, I don't know how YouTube will allow, like, if they're okay with that or if they're not. This is the edge of the sample, and you can see the water is moving a little bit, so don't freak out if you see that big line. It's just water. Yeah, I don't know if I'll get a strike for killing microbes, but probably not, because they're just microbes to people. I know if I was like doing insects and insecticide under the microscope, like I made a video like that a while ago, and people were like, whoa dude, this is probably not a good idea to have up, because the insects did not look like they're having a good time. So, but with I mean, the deaths could be off screen, or it could happen live. Like they don't have faces, like they're not as easy to relate to. I don't know, I'll have to, but I think there are some that are already out there, so yeah, I'll just have to do some searching. Like if they're already out there, then maybe I can just do it. Okay, so I'm just scrolling around. Not finding anything. It's kind of like a night on social media, you know? You just scroll and scroll and you don't really know what you're looking for. And you hope that you're finding something eventually. But so I'm just kind of toying around with some different filters. That's an air bubble right there. Yeah, I mean, if they show rubbing alcohol destroying samples, I guess I could do that. Or I guess I could show the destroying of, because it's like hand soap, and like showing soap killing bacteria is probably like a good educational video. This looks like the remnants of an earthworm. Very small one. Man, not a whole lot of cool stuff on this one. That's okay, you guys. I still have plenty other samples. I have some more stuff from that river sample, and there's lots of green growing in it. So there's definitely going to be some microbes, because like when there's algae, there's definitely, you know, more protists and ciliates. I just wasn't sure about what would be living under the ground. But yeah, it's just lots of dirt and soil. See, I was hoping I'd find like nematodes and stuff. Or water bears. Because like I was Googling like what lives underneath the soil. And they're like, yeah, that's where the tardigrades are, that's where the nematodes are. And, this has been in a watery state for a couple of days though, so there's a possibility that there were nematodes and worms and water bears. Well, water bears live in water, and that's normal, and nematodes are normally fine in water, but I don't know. I'm gonna try doing another sample. Yeah, chlorine eliminating germs. Yeah. I mean, like, I can still... There's nothing that they can do to me, as a channel. Like, I'm not even, like, a monetized channel or anything yet, so even if I got a strike, like, I would just take it down, and you can have up to 3 strikes, I think, for inappropriateness. Anyway, I was showing you guys, this is my next sample that I have here. This was from the river from earlier, and it is... It reeks. It smells like sulfur, like so bad. Here, let me collect some of this. Okay. It'll just be a second, you guys. Hold on. Be patient, be patient. Don't leave the stream. Something really cool is about to happen. Your mind is gonna be blown. To anyone that's been watching the stream, can you just testify and just say, hey, yo, this stream has been amazing, and while Jordan's setting up this next one, just be patient. Okay, thank you for doing that. If you're not, what am I even paying you guys for? Just kidding, I'm not paying anybody here. If you thought I was, then we need to have a talk. Like I said, this channel's not monetized yet. We're definitely a long ways from it. Which I think would be cool to have eventually, but the reason I do this is for the fun of the game and the fun of the hobby, so. And I have another full-time job, you know, so. Okay, almost ready with this sample. I just have to get some stuff cleaned up. I have two, where's the screen? I have two glass slides on here. You guys are getting paid? Yeah, whoops. Oh, whoops. Both of those glass slides have just slid together, so now I just have one big mega sample. Okay, here we go. This is from the Jordan River in Utah, which comes out of Utah Lake, and it used to feed into the Great Salt Lake, but now it's not currently feeding there. It's not high enough. So, let's zoom in here. And I'm pretty sure what we were looking at here is a bunch of cyanobacteria. And I think specifically it is called oscillatoria, maybe. Yeah. There's a rock band called Volvox. That's really cool. I've been wanting to do like some episodes on my channel about microbes in culture and in the media. One is music, because I know that there's music that references different microbes, and I guess there's a rock band as well. Just finding out. But another thing that I've wanted to do is do an episode like video games, like microbes in video games, and video games inspired by microbes or video games that feature microorganisms, because there are a ton, and like there's a whole Wikipedia page about it, and there's like hundreds of video games that have really cool uses of microbes, and like some are like space fighting games, and yeah, but I thought it'd be cool to like collab with like a video game YouTuber or something to do that. I'd have to get like an emulator to like film some of the game footage. It's a whole thing. So all of this cyanobacteria was living on, so it could be algae more related to plants, or these could be bacteria. I'm pretty sure this is a thing called cyanobacteria, and the actual species or genus of it is called oscillatoria. The reason it's called oscillatoria is it gets that name from the word oscillate, which means to move back and forth. And so if you saw this in a like sped up view, you would see them kind of gliding backwards and forwards, and they would normally glide towards the light. In this particular case, the light is right underneath them. So yeah, there's what they look like if they were blue. And here's another view. So yeah, let's see what else we have. Here is some spiral bacteria, kind of cool. Oh, whoops, I think we just lost it. I think some bacteria can be classified as algae, like these cyanobacteria, like if they would be in the same category as algae, maybe not. Sorry, a lot of these things are really small, so I'm like, what should I look at? What should I focus on? It's kind of hard to know. Here's a really small little... let's see, zoom this way. There are some cool little microbes, probably some... I think those are flagellates, like this thing that's kind of next to this green thing. I think it's some sort of flagellate. So difference between a ciliate and a flagellate. Ciliates are like covered in hairs all over themselves. Flagellates usually have like one really long or multiple long tails. With this one it's really hard to see, but I feel like I see one tail kind of longer than another. Oh, now it's connected to some other one. Very interesting. Let's zoom out. Those guys are too small. That's another thing with making these episodes, kind of a behind the scenes thought for you guys is like, there are a lot of living things in these samples, but even with my compound microscope, it is hard to get a good view of everything. And so I'd say probably 40 to 50% of the stuff that I film doesn't even end up making it because it is too small or too grainy, which is really too bad. Yeah, that's kind of the difference between the ciliates and the flagellates. At least that's what I think. This is kind of cool. It looks like a big mouth, which is just the way that I placed it. I was trying to grab at these a couple minutes ago with tweezers, and they just wouldn't grab. And because they're all on top of each other, but it's kind of like pickup sticks. If you try to like grab one, like you'll just grab one. So what I had to do was like I had to scrape it with the tweezers off of the side, and then I pulled it off. I like slurped it up with the pipette. So that's why you get this long strand, is because all of this was just scraped off. Okay, now we're moving to the other slide. This is on the bottom. So it was with the same pipette, so you're gonna see similar things like this cyanobacteria here. Man, where are the water bears? That's what we've all been wondering this whole time. I'll make sure to find some next time. They like to hang out a lot in moss. In Utah, I've only found water bears in one place. Anyone want to guess where I found water bears? Is anybody even here? Is anybody watching? I mean, you guys have still been watching this stuff. Yeah, there's people watching. Water bears, the only place I've found them in Utah is in the mountains, which is kind of funny. It's where you find a lot of the actual bears. So, yeah, no, not in the rain, not in the water. Well, actually, yes, it was in the water. It was in a waterfall. I can't remember the name of the waterfall. It was in a waterfall up in the Wasatch Range in the mountains, and it was in moss. Yeah, and I don't know if it was like the high elevation or if it was just that the quality of the water was better. Because like a lot of the samples that I get are from Utah Lake and the Jordan River, and there's always algal blooms there, and I think that harms the amount of or like decreases the amount of variety that we get. Yeah, I'm gonna zoom over to this other one, you guys. I'm not seeing a lot of stuff. We'll hang out with this cyanobacteria for just a little bit. I think it's called Rocky Mouth Falls, maybe. It's right by Little Cottonwood Canyon, if anybody knows where that is. And yeah, it was some moss that was like underneath like this really tiny waterfall in the middle of spring. So, and I've collected some there, but I've collected moss here in my backyard. I've collected moss near the Jordan River and Utah Lake. Nothing in there. I've collected moss in southern Utah, like in St. George area where there's like red rock, no tardigrades there. Utah is just a really dry place, so I feel like even if there were tardigrades, like their seeds would not do well here for a long time. It's dry and it's hot, so yeah, it is really picturesque. Probably very picturesque for the tardigrades as well. Tardigrades. Oh, let's look at this under the Darkfield for a second. You can't neglect the Darkfield lens. And let's block that out a little bit more. Ooh, so much cooler now. Moss piglet, tardigrades, call them what you want. They probably will never know what you call them. Wait a minute, look at all these little... Oh, you know what those things are? All those little brown spots, I think those are scratches on the cover slip. Because if I zoom out to them, yeah, those are scratches on the cover slip. I was like, wait a minute, we found something new. We did not find something new. That's okay. Yeah, really cool contrast, huh? Yeah, so I think what I'm gonna do is, I think what happened is that, so this sample, you know, I've had since last week, so I think it's just gotten kind of stale, and the cyanobacteria has kind of taken over. So what I'm going to do is I'm going to get some fresh samples for next week, and I'm going to collect some moss, and some people earlier wanted us to look at blood under the microscope. So I think we'll do some of those things, and I'll find another prepared slide thing or two. But yeah, I think we're getting pretty close to wrapping things up here. We've looked at at least 3 different jars, you know, so I think it's time to start wrapping things up. Oh yeah, let's see. Here's a cool little hair here. Yeah, even though there's not like a whole lot going on, like, it's still pretty cool to look at, you know? It's just nice to enjoy. But yeah, I think this is going to pretty much wrap it up for today. I really appreciate you guys for being in here for this viewing. Thanks for participating in the chat. It helps me not feel like I'm talking to myself. Coins and Capsaicin, Bite-Sized Stories, Kay Jesperson, Nader, Erknees, you guys have all been around for, and Daniel Goats, I think I've said that. You guys have all been around for a long time. Thank you so much for watching all of this stuff. Again, I'm going to be here every Tuesday night at 8 o'clock Mountain Time, usually 8 to 9:15, 9:30. And again, keep posting what you think I should look at next, like where else I should get some samples. And then, yeah, I've got the Patreon if you want to help support the cause. Help me get a new objective lens so we can look at bacteria a little bit close up. That's the next thing. Yeah, I've got some other cool stuff coming up. Some kids microscope reviews, some adult microscope reviews, and some other things. So lots more. So thank you all so much for watching. Remember to enjoy the little things in life, and I will see you, or you guys will see me next time. So chlorophyll, can you see it? Yes. We'll definitely try to look at the chlorophyll and some parrot feathers. Yeah, that'd be really cool. Yeah, keep posting some stuff in the chat, and we'll see. But yeah, anyway, enjoy the little things in life, and you guys will see me very soon, and I hope to see you in the chat very soon. Anyway, later.